Aloha! Today I will begin presenting the last stage of cellular respiration, mainly the pathways of oxidative phosphorylation. I will also talk about photophosphorylation, but since photophosphorylation is part of photosynthesis and photosynthesis is not included in our curriculum, I will only define it and then I will leave it there. Alright, so I will start by asking these questions. What is the goal of these pathways? Oxidative phosphorylation and photophosphorylation. The objective of these pathways is to generate ATP. As you know, ATP is the energy currency in cells. All processes are made possible with the help of energy contributed by ATP hydrolysis. And by now you already know that it's not simple ATP hydrolysis. It's the energy transfer as a result of breaking down high energy bonds in ATP. The energy then is invested in whatever purposes that energy is needed for, but it's not simple hydrolysis. Alright, so where do these processes or pathways occur? They occur in the mitochondria. So simply put, what happens during these pathways, these processes? Now normally when I present this you don't see the whole slide, but it doesn't really matter. The information is here anyway, and so I'll just, if I happen to read some parts straight from the slide, just don't count it against me. So oxidative phosphorylation, this is a process of reduction of molecular oxygen to water. And the process is stimulated by electron flow from electrons donated from various electron carriers, but mainly NADH and FADH2. The oxidative phosphorylation pathway occurs independently of light. This is going to be the pathway, or the pathways we're going to focus in chapter 19 primarily. Now, photophosphorylation on the other side is a light-dependent oxidation of oxygen from a water molecule that will be converted into a molecular oxygen with NADP plus as an electron acceptor. So in a way, this is slightly, well, it almost appears as if it is the opposite process. In the oxidative phosphorylation, we oxidize molecular oxygen to water. In photophosphorylation, we oxidize the water oxygen to molecular oxygen. But the commonality between them is that in both processes, the goal is to generate ATP. Regardless of how that is achieved, the overall goal is ATP generation. So how do electrons facilitate ATP synthesis? This is also known as the chemiosmotic theory. And the way the electrons provide the energy for these processes to occur is actually quite similar between the two pathways. So they both involve electron flow. And the energy from the electron flow is involved in the movement of protons against their concentration gradient, thus generating an electrochemical gradient across a semi-permeable membrane, in this case the inner mitochondrial membrane. That gradient then will be used to drive the synthesis of ATP. So that is similar for both processes. So obviously the mitochondria will play a major role in this process and in the energy production. That's why they are known as the energy organelles in the cell, energy producing organelles in the cell. Alright, so anatomically speaking, the mitochondria is made out of outer membrane, which is fairly permeable, especially for small molecules, ions, etc. But then underneath is the inner mitochondrial membrane, and that membrane is very selective, and not many things can pass through it. I mean, even protons are not allowed to pass through it. Something characteristic for the inner mitochondrial membrane is that it forms these digitations or convolutions or cristae, also known as, and the purpose of these is to increase the surface of the inner mitochondrial membrane, and that is the surface where we will find the electron transport complexes, also the ATP synthase, and some of the transporters that facilitate passing of ions and other molecules across the inner mitochondrial membrane. And then inside is the matrix that contains all the enzymes involved in the citric acid cycle, beta oxidation, amino acid oxidation, many other metabolites, some DNA and ribosomes, etc., etc. So that's the structure in general of the mitochondria. Now, when we talk about electron transfer, there are really three main mechanisms of transferring electrons from one carrier to another. The first type of transfer is through a direct contact or direct transfer. Example is the reduction of the ferric ion to ferrous ion. Another way to transfer electrons is by transferring a hydrogen atom. We have a proton that contains an electron that's basically a hydrogen atom. The hydrogen atom transfers one electron. And the third way to transfer electrons is by transferring a hydride ion, where the hydrogen atom carries two electrons and they are passed to whatever is their next destination or the next acceptor. Alright, so what is the name of the electron transfer reaction? I mean, all of you know the answer to this question, but I like to start logically. Let's think about it. Obviously, we have a reduction and oxidation, so the name of the reactions are oxidation reduction or redox reactions. So how many different electron carriers have we come across so far? I mean, we've talked about NAD+ and NADH, which are usually involved in degradation processes, and something very typical for them is that they're reversibly associated with the redox enzymes, redox reaction enzymes that usually carry the name dehydrogenases. We also have heard, even though not very often at this point, about NADP plus and NADPH electron carriers. They're primarily involved in anabolic processes. We've talked about them in the pentose phosphate pathway though, and also in the process of fatty acid degradation, the polyunsaturated fatty acids, if you remember. These electron carriers are also reversibly associated with their enzymes. The other electron carriers we've talked about are primarily FAD, FADH2, and I don't know whether we've mentioned FMN and FMNH2 at this point, but if we haven't, we will. So these are very similar in structure, just slightly different. Something characteristic about these carriers, we've talked about them already, is that they're associated with their proteins, but not transiently. They're permanently associated with their proteins, and their oxidative potential is determined by the protein with which they're bound to. Something else that makes these unique is that these carriers can accept either 1 or 2 electrons, whether the nicotinic electron carriers can only accept 2 electrons at a time. Alright, so in addition to these electron carriers, that most of which we've talked about before, there are a few other types that we are going to talk about when we talk about the electron carrying pathway in the oxidative phosphorylation processes. One of those is ubiquinone. We also know it as coenzyme Q or just Q. Now this is a benzoquinone that is lipid soluble, and it can freely diffuse in the lipid bilayer. And it can accept 1 or 2 electrons. It can be either in a fully reduced form as, fully oxidized form as ubiquinone. It can be in the semi-oxidized form and containing 1 extra electrons, and as such is actually a radical, and that's semiquinone, or in a fully reduced form known as ubiquinol. So the complete reduction of ubiquinone requires 2 electrons and 2 protons, and it does occur through these 2 stages of semiquinone and fully reduced ubiquinol. Another type of electron carriers that we are going to mention in our future studies are the cytochromes. We have 3 types, A, B, and C. Cytochromes are heme-containing proteins which, based on the fact whether they are oxidized or reduced, have a different Lip's absorption spectrum. And then I would normally ask you, is this property useful and how? And I would hope that you would realize that if we can distinguish between oxidized and reduced state of the cytochromes, we could use that to study metabolic progression of metabolic reactions. You can set up a reaction in a spectrophotometric cuvette, and then start the reaction and measure how much the cytochromes are either oxidized or reduced. And by that we can determine rates of reactions, we can determine if reactions occur, if we use inhibitors, how they affect, which stage, which step. So this is invaluable. This is highly useful research tool. Okay, so cytochrome A and B and some of the cytochrome C's are actually integral proteins, but the mitochondrial cytochrome C is a peripheral soluble protein. It means it's not permanently attached to a protein complex, and we'll see that when we talk about complex 3 and 4. Also, cytochrome C has a covalently attached heme group. This is the structure of the heme group that is part of the cytochromes. You've seen a similar structure when we've talked about hemoglobin. Hemoglobin also contains a heme group that is made, each heme group basically has a porphyrin ring, and each porphyrin ring is made with these 4 5-membered rings known as the pyro rings, and their structure basically helps to stabilize this iron right in the middle, and this iron can be either in the ferrous or in the ferric state. In hemoglobin, this iron can only be in the ferrous state, F2+, Fe2+, but here they can be either in Fe3+ or 2+ form. And you can see here that cytochrome C in the mitochondria is, actually cytochrome C is period. The heme group is permanently attached by 2 cysteine residues to the protein part of the cytochrome molecule. All right, you don't have to try to remember any other distinctions between the three cytochrome types, but one thing that distinguishes cytochrome C is that the heme group is covalently attached to the protein. All right, this graph shows how the two states of each cytochrome differ by these three peaks known as alpha, beta, and gamma. In this particular case, it's only shown the cytochrome C, but they all have a similar pattern. The reduced form that has only one peak, the gamma peak, and the oxidized form has only one peak. The reduced form has three peaks, alpha, beta, and gamma. Now, if I were to ask you which wavelength would you use to distinguish between the oxidized and reduced form, what would you say? Many of you, when I ask this question, choose the wavelength around 400 because that's the maximum absorbance of the cytochrome C. But that is the wrong answer. You don't want to choose 400, which is the wavelength of the gamma peak, because at that wavelength both form absorbed at maximum intensity. But you will say, but there is a difference between them. Yes, indeed, there is a difference. But if you have 75 or 80% maximum absorbance for the oxidized and 100% maximum absorbance for the reduced, the difference is only 20%, but that's at maximum absorbance. If you transition from oxidized to reduced or reduced to oxidized, you're transitioning from one maximum absorbance state to another maximum absorbance state. It's going to be very difficult to distinguish the contribution of each state because they overlap. You want to choose a wavelength, even if the absorbance is not great, where we have a very distinct difference between one and the other state. And that wavelength is actually the wavelength of the alpha peak. In this particular case for cytochrome C, that wavelength is about 550. Why is that a good choice? Even though we only have such about like 20-25% of relative absorption for the oxidized form. Well, even if the absorption is not that great for the reduced form, it's almost inexistent for the oxidized form. So there is a very distinct difference between the two states. You can clearly see the transitioning from oxidized to reduced and reduced to oxidized. Now, the wavelengths of the alpha peaks are used to distinguish between the three types of cytochromes. The alpha peak for cytochrome A is around 600, for B is about 560, and for C is about 550. So based on which cytochrome we use, we have to use the appropriate wavelength if we want to distinguish between oxidized and reduced forms of these cytochromes. For example, you cannot use 600 to distinguish the difference between oxidized and reduced forms of cytochrome C. I mean, they both will have almost no absorbance at 600. Even at 560, they will have pretty much the same absorbance. You're not going to get anything. So it is critical that you use the appropriate wavelength for the specific cytochrome in order for you to get a reliable data. All right, the third kind of electron carriers that we are going to talk about during the oxidative phosphorylation pathway, so electron carrier pathways, are the iron-sulfur proteins. These proteins do not contain a heme group, even though it says iron-sulfur proteins. These proteins actually do contain iron, but that iron is associated either with elemental sulfur or with the sulfur from cysteine residues. And in one case, in the case of risky iron-sulfur proteins, it's associated also with histidine residue. And these proteins can only transfer one electron. And the cytochromes, to be honest, they could transfer 1 or 2 electrons, like it says here. Cytochrome C specifically transfers 1, but the others can either transfer 1 or 2. Okay, this is the representation of the iron-sulfur proteins. You can see iron here associating with the sulfurs from 4 cysteines. Again, a more complex structure where we also involve elemental sulfur or some more complex crystal-like structures. All right, so the order of electron transfer has been determined to be from NADH or succinate to flavoproteins to ubiquinone to iron-sulfur proteins to cytochromes to oxygen. Now that's in general. This doesn't mean that you might not see in some cases iron-sulfur proteins, for example, earlier in this chain of events. But in general terms, that is the order. I guess a good question would be, how do we know that order? And, many of you, especially when looking at this table, would say, well, that's because of the reduction potential. And the higher the reduction potential, the more likely is for that compound or structure to accept electrons. And then I would normally ask you to design experiments which can help you determine whether the electron flow truly follows this order, either from NADH or succinate to flavin proteins to ubiquinone to iron sulfur to cytochromes and to oxygen. How would you do that? And I usually will allow you to think about it, but for the sake of time, and since you're not in an in-person class, I'll just explain it. So the way this is experimentally determined is basically by trial and error. You start mixing different things in a setting that mimics as much as possible the settings of a mitochondria. And then by use of different inhibitors, you can try to determine the sequence of events. For example, if you block the transfer of the electron carrying pathway between NADH and Q, none of these other following members of the electron carrying pathway would be reduced and oxygen will remain. Now, if you use antimyosin A that blocks the transition between cytochrome B and C1, well, you will get reduced and oxidized. Actually, you get electrons will pass from NADH to Q and from Q to cytochrome B. So cytochrome B will be reduced, but they will not pass to C1, C, and to oxygen. So there will be a block right here. Electrons will accumulate in the cytochrome B form, and they will not reduce the cytochrome Cs and the oxygen. And using the same logic, we can block various pathways, including the last one with cyanide or carbon monoxide, to really prove and establish the actual sequence of events that occur in the electron transport or transfer pathway. Okay, so this will be the last slide for today. I just want to introduce the members of the electron transfer pathway, but we will look into in more details the exact reactions that occur in these complexes. So 4 enzyme complexes are involved in the electron transport from the electron carriers NADH and FADH2 to oxygen. And they are located in the inner mitochondrial membrane. Complex 1 is the NADH dehydrogenase. You will understand the meaning of the name a little later, but you can tell that you're obviously going to oxidize NADH. This is a massive complex, 850 kilodaltons. It has 43, and in bacteria 14 subunits. Some of the prosthetic groups: iron, sulfur, FMN. Second complex is succinate dehydrogenase. This name should be familiar to you. You should be thinking about the TCA cycle. So you can see that the TCA cycle, a member of the TCA cycle, is also a member of the electron transfer pathway. Again, very large complex. The next one is ubiquinone, cytochrome C, oxidoreductase. And the last complex is cytochrome oxidase. Now, the way the electrons flow is complex 1 will pick up electrons from NADH2, and complex 2 will pick up electrons from succinate, or FADH2, and these electrons will be transferred to ubiquinone, converting it to ubiquinol. Then complex 3 will take the electrons from the reduced ubiquinone, known as ubiquinol. It will take the electrons and it will load them up on cytochrome C molecules, which will travel to complex 4, and where the cytochrome C will donate its electrons ultimately to molecular oxygen, converting it to water. Now, during these transfer process, not in every stage, but in most stages, protons will be moved across the inner mitochondrial membrane into the intermembranous space between the outer and inner membrane. And these protons will be moved against their concentration gradient. This is going to be an active transport. And then once that gradient is generated, then later on it will be used by ATP synthase to generate ATP. All right, that's all I wanted to share with you today. If you have any questions, please let me know. I will be happy to answer anything you have at this point. Thank you for your time.